Industrial production of microbial fermented foods like bread and cheese, and fermented vegetables like olive and cucumber. Bread. Bread is a dietary product obtained from the fermentation and subsequent baking of the dough, mainly made up of cereal flour and water. Made in many different ways, sometimes enriched with typical regional ingredients. Um, the bread are in so many types like white bread, brown bread, rye bread, and wheat bread. The ingredients of the bread: flour, yeast, sugar, salt, water, fats and oil, and starter culture. These ingredients are mixed together in the bread making process. General bread making process include these steps: mixing, raising, kneading, molding into loaf shapes, rising, baking, cooling, slicing. The first step is mixing, distributing the ingredients eventually. After addition of the yeast, there will be little growth or no growth in first 2 hours. After 2 to 4 hours, there will be a growth, and 4 to 6 hours decline in growth. There are no growth in the medium. During fermentation, conditioning takes place when flour protein, that is gluten, will mature in that period and become elastic. The elastic, stretchy thing is because of the gluten. The rate of gas production increased by the yeast by adding more yeast, and second one is sugar. Decreased by the addition of salt or too or low high temperature. The fermentation room temperature will 27 degrees Celsius. Eliminating by chemicals. Carbon dioxide gas directly incorporated to dough with the baking powder. Ammonium bicarbonate will release carbon dioxide, ammonium gas by heating. Flavor production. Alcohol, acids, yeast are added to this. The baking must be in 100 degrees Celsius, that will kill the yeast and inactivate the enzymes. After the baking, the cooling process will happen. The whole load is cooled to about 35 degrees Celsius before slicing and wrapping. The second one is cheese making process. A food consists of coagulated, compressed, and usually ripped curd of milk separated from the whey. There are so many types of cheese are present in the world. For example, cheddar cheese, mozzarella cheese, cottage cheese, blue cheese, etc. Cheese making process. Cheese consists protein and fat from milk. Usually the milk from cow, buffalo, goat, or sheep. It coagulates by the milk protein casein. The milk is gradually heated to 30 to 35 degrees Celsius before acidification and coagulation. There are some steps in here. Acidification. Starter culture is added to the milk, change lactose, that is milk sugar, into lactic acid. This process changes the acidity level of the milk and begins the process of turning milk from liquid into solid. The next is starter culture. Fermentation starters are precipitation through acids, the beginning of the fermentation process in preparation of various food and fermented drinks. A starter culture is microbial culture which actively performs fermentation. Coagulation. Rennet is added to the further coagulates to the milk to solidify, forming a custard-like mass. Rennet, an enzyme used to coagulate milk during the cheese making process. Rennet is derived from one of 4 sources: the stomach lining of young calf, plants, Microbes in fungal and yeast, generally engineered rennet that imitates animal rennet. The next step is cutting. It is then cut into small pieces to begin the process of separating the liquid that is whey from the milk solids that is curd. Large curds are cooked at low temperature, yielding softer cheese like mascarpone, ricotta. Then stirring, heating, drying. Cheesemakers cook and stir the curds and whey until desired temperature and firmness of the curd is achieved. The whey is then drained off, leaving a tight, firm curd. The next step is salting. Salt added flavor also act as preservative so the cheese does not spoil during long months or year of aging. It also helps a natural ring to form on the cheese. Next step is curd transformation. Different handling techniques and salts affect how the curd is transformed into many cheese varieties made. Next step is shaping. The cheese is put in a basket or mold to form into a specific shape. During this process, the cheese is also pressed with weights or machine to expel any remaining liquid. Pressing determines the characteristic shape of the cheese and helps complete the curd formation. Most cheeses are pressed in 3 to 12 hours depending on the size. Aging should take place in controlled environment. Different cheeses require different temperatures. Humidity, however, in a small refrigerated temperature is kept at 55 Fahrenheit at 85% of humidity. During aging, the cheese should be rotated or flipped periodically to prevent the moisture from the setting in the cheese and prevent the inconsistent internal consistency. Now we will move on to the fermented vegetables like olive. The main use of raw olive is as olive oil. More than 90% of the total worldwide olive production is used for oil. 7% to 10% are consumed as table olives. Currently, 4 countries, Italy, Spain, Greece, and Turkey, are responsible for 75% of the total worldwide olive production, 9 to 5 billion kilograms. In table olive fermentation, phenolic consistency, reducing sugar content, microbial profile, salt concentration, acidity, and temperature are the main parameters that should be considered for providing a proper fermentation conditions. Table olive fermentation is a complex biochemical process involving different microorganisms. The microbial composition of affected by several factors such as composition of the brain, ambient acidity, salt concentration, olive variety, olive phenolic content, temperature, oxygen availability, alkaline concentration during the fermentation. Mainly microorganisms that can be found in the fermentation medium are Enterobacteriaceae, lactic acid bacteria, and yeast. However, yeast and lactic acid bacteria are the main role in the fermentation process. The main lactic acid bacteria species involved in the olive fermentation are Lactobacillus plantarum and Lactobacillus pentosus, and yeast species Saccharomyces cerevisiae. Fermentation. The initial pH fermentation can be about 7 depending on how much washing was done after the NaOH treatment. Each species may be present and contribute desirable flavor characteristics to the brained olives. Olives are placed directly in brain with a salt concentration of about 6% to 10%. The fermentation process can last 8 to 12 months and it's mainly driven by yeast and lactic acid bacteria. The second vegetable is cucumber. Botanically, cucumber from the same family as watermelon and pumpkin. Pickling cucumbers. Compared to slicing cucumbers, pickling cucumbers are usually shorter, 3 to 4 inches long, thicker, less regularly shaped, and have pumpy skin and tiny white or black dot spines. Color can vary from creamy yellow to pale or dark green. Commercial fermentation is considered in open air tanks, and open pouring is applied during the active fermentation period. Raw materials preparation. Select the raw materials, that is, select cucumber without brushing or damage. Wash the cucumber in potable, cold water and drain. It's important that the water used for the washing is clean. Boiled water is still to avoid the contamination of the brain by waterborne bacteria. Chlorinated water should not be used for washing the fruit this could prevent the natural fermentation taking place. The cucumber are sliced into thin round slices. Mix the salt with cucumber. This allows the brain to form by osmosis. This takes few hours depending on the temperature. Add vinegar and finally the fermentation occurs. As soon as the brain is formed, fermentation starts at 21 degrees Celsius and bubbles of carbon dioxide will appear. Fermentation takes place between 1 to 4 weeks depending upon the ambient temperature. Fermentation is complete when there is no bubble, it'll appear. And that's all. Thank you.